So today we are going to look at a very important topic, that is macromolecules and biosynthesis. So today we'll look at the part 1 that has to deal with carbohydrates and lipids. So these are very important molecules that make up our body. They have some great importance in our body that we are going to see as we proceed. So first of all, we have to note that living beings are always made up of a limited number of atoms which combine together to form molecules. So these molecules range from very little molecules like carbon dioxide, oxygen, water, to very huge molecules that are very huge, like proteins, lipids, nucleic acids, and some carbohydrates. Molecules that are very huge, and at times some of these molecules can be visible to us humans with the help of a microscope. So you could imagine how big these molecules are. So we call these molecules macromolecules. Macro, which means large. So these are large molecules. Okay. So these smaller molecules, they are easily mobilized and they are more soluble. Okay, they tend to be used for cellular activity like metabolism, like carbon dioxide, for example, which is a waste of metabolism. You have oxygen, for example, which is being used for metabolism, and so on. So macromolecules, however, they are used for storage. So they have functions in storage. They have functions in the structural constitution. So an example here, for example, production of membranes, production of cell walls, and all of the rest. So structural constitution of cells. And in some other cases, like in the case of DNA, they have purpose in storing information. So they are informational molecules, right? They possess genetic material. So here we have to talk of nucleic acids. So a macromolecule is mainly made up by addition of little monomers. That means all of them, they are made up by monomers that come together in numbers to form these very huge molecules. Okay, so in what we call a polymerization reaction, and majority of these polymerization reactions are dehydration reactions, or at times we can call it condensation reactions. We are going to see that these condensation reactions have different names for the different molecules. The bonds that are hence formed also have different names, which you will all know by the end of this. So for example, here we see you have a short polymer. So you have 1, 2, and 3 monomers that make up this short polymer. They combine together through a dehydration reaction or condensation reaction with the loss of water, and another monomer unit is being added so that you now have a longer monomer with 4 monomer units. Okay, so this is an example of the production of a macromolecule, how we can produce macromolecule from smaller, simpler molecules. An example of breaking down a macromolecule or a polymer is by hydrolysis. Hydrolysis, which means we are doing the reverse. We are no more removing water, but instead we are adding water. So we add water to the polymer, to the bond in the polymer. We said we are going to look at the names we give to these different bonds for the different macromolecules. So we add water to these polymers, and at the end we have the molecules, the bond that is being broken down, and the molecules that is being separated, and you have the monomer units that reduce. So let's start by looking at the carbohydrates. So the word carbohydrates come from compounds that are made up of carbon and water. Water that is made of what? Hydrogen and oxygen. So these compounds, they contain carbon and hydrogen with oxygen. So it is very important, as the name says, hydrate water, to note that the ratio of hydrogen to oxygen is always 2:1. Okay, like in water. That is why it carries the name hydrate. So they all present the general formula CxH2O(Y). We note here that H2O is a formula for water. So CxH2O(Y), where X and Y are variables, so they can change. X can be 3, Y, for example, could be 3 or something else, depending on the different carbohydrates. But all of them respect this general rule. So their chemical reactions. So you have to know that the chemical reactions are defined by the different chemical groups that they possess. Okay, so some of them have the aldehyde functional group, and whereas some of them have the ketone functional group. So these, for example, they can take part in some particular reactions. For example, carbohydrates that have the aldehyde functional group can enter some particular reactions that carbohydrate with the keto functional group cannot enter into. So they have difference in the chemical reactions that they undergo due to the functional groups that they possess. Also, all of them have hydroxy groups, and these hydroxy groups, depending on their position, they can have different properties. So what you should all know is that their chemical reactions are defined by the different chemical groups that they possess. Okay. So now we are going to say that, and you all know that carbohydrates are divided into 3 main groups. That is monosaccharide, disaccharide, and polysaccharide. So let's start with monosaccharide. So monosaccharides, they are the simplest forms of carbohydrates. They serve as the building blocks for larger carbohydrate molecules. So we use monosaccharides just as monomers, as monomer units, as we were looking at the monomers previously. So we use monosaccharides, we merge them together, we bond them together to produce longer chains of polysaccharides. So they are the building blocks of larger carbohydrate molecules. Also, they exist as single sugar units. They are very sweet and they are highly soluble due to their little size. So they have general formula CH2ON, and they can be classified based on the number of carbon atoms that they present. So if they present 3 carbon atoms, we call them trioses. Oses, which has to deal with carbohydrate. Okay, so oses is another suffix to say that we are actually talking of a sugar. Okay, just like glucose contains O's, just like galactose contains O's, just like sucrose contains O's. So that O's is just a suffix to indicate to us that we are talking of a carbohydrate. So if they contain 3 carbons, we call them trioses. If they contain 4 carbons, we call them tetroses. If they contain 5 carbons, we call them pentoses. If they contain 6 carbons, we call them hexoses. So examples of trioses: carbohydrates, monosaccharides that contain 3 carbon atoms. We have glycerate, we have acetone. So those are examples of 3 carbon containing monosaccharide. Then you have for tetroses that contain 4 carbon, 5 carbon. So we're going to look at all of that in detail. So they have the presence of either an aldehyde group or a ketone group, as we previously said. So if they have an aldehyde group, we are going to call them aldose sugars or aldoses. Remember, "-ose" is a suffix to indicate we are talking of a sugar. So if they contain the ketone group, we will call them keto sugars or ketoses. So let's look at the different monosaccharides and their function. So we previously mentioned that there were trioses. We gave examples of glyceraldehyde, glyceric acid. Glyceraldehyde, 3 carbon atoms, and it contains an aldehyde functional group. Okay, so this is an aldose sugar. Then you have dihydroxy acetone. That is a sugar with the very same number of carbon atoms, but now with the ketone functional group. So this is a ketose sugar. Okay, so these trioses, they mainly serves as intermediate during the cellular respiration, glycolysis, and they also help in photosynthesis, that's in the dark reactions. Then you have pentosis. Pentosis, example of pentosis, you have ribose, you have deoxyribose, you have ribulose. So ribose and deoxyribose, they help in the constitution, synthesis of nucleic acid. Ribulose helps us, it is a carbon acceptor in the Calvin cycle, which all of you certainly know. So some of them also help in the synthesis of coenzymes like NAD, fat, and also in ATP synthesis. Okay. Then there are hexoses, which are sugars which contain 6 carbon atoms, as we said. So among these sugars, we can state glucose, fructose, and galactose. So these are the 3 main hexose sugars. So these hexose sugars, their functions are mainly as a source of energy. So they are mainly used to produce energy and to synthesize larger molecules, disaccharides and polysaccharides. So the most common monosaccharide is glucose, as you must have noticed. And the simplest monosaccharides are glyceraldehyde and dihydroxyacetone. So this is a structure of an aldose sugar or an aldehyde sugar. This is a structure of a ketose sugar. Okay, so you can observe here keenly that we have 1, 2, 3 carbon atoms, and we have here what? We have here this carbonyl functional group, which specifically is what? An aldehyde. So you have an aldehyde, a terminal carbonyl group. So you have an aldehyde. Whereas here you have the same number of carbon atoms, same number of hydroxy group, everything is the same, but apart from the functional group. So here the carbonyl group is intermediate, so this is a ketone. So you have an aldose sugar here and a ketose sugar. So glyceraldehyde and dihydroxyacetone. Then looking at the hexoses, if you take glucose for example, look at glucose. Glucose has 5 OH groups and a terminal aldehyde functional group. Okay, galactose has still 5 hydroxyl functional groups and an aldehyde terminal groups, but now the difference comes at the level of the spatial arrangement of these molecules. So as you see, the difference is at the level of this third hydroxyl group, which for glucose is found at the left and at the right, and for galactose is found at the left. Then fructose is almost the same, just that for fructose now you have a different functional group, which is a ketone functional group. So fructose fall under ketose sugars. Okay, so we saw that there are 2 main ways of classifying these sugars. One, according to number of carbon atoms that they have, as either trioses, tetroses, pentoses, or hexoses, or also according to the functional group which they have, that's either as aldoses or ketoses. Now let's look at what a disaccharide is. So disaccharides, they consist of 2 monosaccharides units that are joined together by glycosidic bond. So remember, we said that these polymers were formed by the merging of many monomers together, and that those monomers, they merge through a dehydration reaction, or we called it also a condensation reaction. So that condensation reaction that releases water causes the production of a bond in between the monomer units. So a new bond is being formed, and that bond in the case of carbohydrate is called a glycosidic bond. To each and every one of you should note it. So very important. So this here is a glycosidic bond. So we said they are formed by dehydration reaction between the hydroxyl group of each molecules, and you have the formation of water. Okay, so the bond is usually between carbon atoms 1 and 4, and they are usually formed from hexoses. Okay, so at times hexoses, or most of the time naturally, these hexoses are found in the ring form. So they have the aliphatic form and they have the ring. That means they have the normal straight chain form and they have the ring form, and normally in nature they are found in this ring form, which we are going to see. So the most common disaccharides include maltose, which is made up of two glucose monomers that are bound together by a glycosidic bond; lactose, which is made up of still two monosaccharide units, that's glucose and galactose, linked still by a glycosidic bond; sucrose, that is made up of glucose and fructose. So, and sucrose is the most abundant disaccharide in nature. Okay, this sucrose is is mainly being found in plants, in sugarcane for example, and in fruit, and some fruit. So remember I said majority of these hexoses, they are being found in the ring form. So this is their ring form. So you see here that they are in their ring form. You have what you call you have the carbon atoms. So in the ring form contains oxygen. Okay, that is in the ring itself. You have oxygen in the ring itself. So we start counting the carbon atoms from here. So you have carbon atom number 1, 2, 3, 4, 5. Okay, so here let's repeat it: 1, 2, 3, 4, 5, and this carbon atom is carbon atom number 6. So the glycosidic bond, you should note, is formed in between carbon 1, right, of this glucose, and carbon number, and the OH at carbon number 4. Okay, so the OH at carbon number 1 and the OH at carbon number 4. So that is why we see the glycosidic bond is formed, but it is you have the 1,4 glycosidic linkage. We talk of the 1,4 glycosidic linkage because it is in between carbon atom number 1 and carbon atom number 4. 1,4 glycosidic linkage. Very important that you know. So this is the structure of maltose. Okay, good. Then for the production of sucrose, it is almost the same thing, just that now the difference. So this is for the production of maltose. For sucrose, now you have a different linkage. So what you should know mainly is that of glucose, because that is what they will mainly ask. For the rest, you should just know that the glycosidic bond is in between the OH and the H, and you have a condensation reaction and it links the two monomers or the monosaccharide units together. Now polysaccharides. So polysaccharides, these are even larger molecules that are made up of multiple sugar units. So you have more than 10 units. So you have a lot of monosaccharide units that make up polysaccharides. These polysaccharides, they are very, very large. They are mainly the storage forms of food in our body, of carbohydrates in our body. So there's the storage forms, which are mainly starch and glycogen. So starch, as you must know, in plants, and glycogen, as you must still know, in animals. So they also help in a structural material, example cellulose. Okay, that help as structural material. So they have some structural function. Okay, so these polysaccharides, they are convenient storage molecules due to their large size, which make them very insoluble in water. So they exert no osmotic or chemical influence on the cell. Okay, they can easily be converted to simple sugar when there is need. Then you have also some other complex molecules that resemble the polysaccharides. You have chitin in fungi and some arthropods, and murine in bacteria. So this is, for example, is the structure of chitin. Okay, this is the structure of chitin. Not very important. So as you see here, the The surgical threads that we use in the hospital are made up of chitin. Why? Because this chitin has a property. It has a property of dissolving, of being used by our own body. So when we use it as thread, for example, to sew a wound or to sew something in the body, in fact. So this thread will not remain in the body forever. Why? Because the chitin which it's made up of is a kind of it's a monocyte, it's a metabolite, so it can actually be used by our body, and it is being dissolved. So after some time it is being dissolved. Okay, after the wound has healed, it's being absorbed back by the body. So now let's look at lipids. So lipids are heterogeneous group of hydrophobic molecules. Okay, so heterogeneous meaning that you cannot bring a general, there is no general formula for lipids like there is for carbohydrates. Okay, they are heterogeneous. We're going to say that there are many types of lipids. So lipids, they are major sources of energy for the body, and they equally serve as hydrophobic shield against foreign substances that try entering the body. So due to the high chemical variability, lipids cannot be defined accurately. So we said it is a heterogeneous group, so we cannot define it accurately like we did for carbohydrates, saying that carbohydrates were formed, they were organic compounds that were made up of carbon and hydrogen and oxygen only, in which hydrogen and hydrogen had a particular ratio which we stated. So for lipids, we cannot give a precise, accurate definition. Why? Because they're a heterogeneous group. They are very broad. You have some compounds that are very different from each other. So they are therefore loosely classified as being formed by condensation of fatty acid with an alcohol. But you see that this is not always true. Okay, that was the definition we give in ordinary level and advanced level. So the building blocks of lipids are fatty acids. Okay, they have a general formula of R. So this is a general formula of fatty acids. Okay, of RCOH. So they are like carboxylic acid. So fatty acids, where R can be hydrogen or any organic group such as a methyl group or an ethyl group and so on, increasing by addition of a methyl group as the series unfolds. So you have always a terminal carboxylic acid, and then you have R, that is an alkyl group that is very long, so we call them fatty acids. Okay, so naturally occurring fatty acid presents an even greater number of carbon atoms. So they have a very great number of carbon atoms that can be at time 18, 21, a lot of carbon atoms. So they are a very long chain. That's what makes them insoluble, because the alkyl part, which is the longest part, is insoluble. So the characteristically long tail of carbon and hydrogen atom in fatty acid consists of lipid hydrophobic tail. So hydrophobic meaning they don't like water, so they don't dissolve in water. Okay, so many lipids properties are determined by those tails, especially their insolubility in water, as we said. So fatty acids with one or more double bonds are termed unsaturated. So if a fatty acid has a double bond or more, we call it unsaturated, while those without any double bond, those that have no double bond, are called saturated fatty acids. So some common fatty acids that are in man, you have stearic acid with 18 carbons, you have palmitic acid with 16 carbons, you have oleic acid with 18 carbons. There exist two types of fatty acid based on the endogenous synthesis. So you have endogenous, that means the synthesis in our body. So you can classify mainly as essential fatty acids. So essential fatty acids, so they are called essential. Why? Because they cannot be synthesized in our own body. Okay, so the synthesis for these fatty acids cannot be done within our organisms. They can only be gotten from our diet. Examples of this, you have linoleic acid. Okay, it's an example because the body does not know how to synthesize it, or rather it does not have the qualities or what is necessary to synthesize these fatty acids. That's why they are essential. The only way for us to get them is through our diet. Then the next group of fatty acids are the non-essential fatty acids, which are non-essential because our body already has the resources to synthesize them. So lipids are roughly classified as, we can also classify them as triglycerides. We are going to see this, very important, triglycerides, as phospholipids, as steroids, and glycolipids. So these are the main 4 important classifications of lipids which you should all know. Good. So for example, here you have a diagram that shows what, so this here is a fat molecule, okay, a lipid. We have a fat molecule, so we call it a fat. Why? Because it is very long and it is solid. So they are lipid molecules that are solid, that's why we call them fat. Then oils, they are liquid. Okay, so fats are saturated. Fats are saturated, okay, and majority of these oils are unsaturated. Good. So example of a fat, you have butter and all of the rest. So this fat, as you might see here, you have just a normal chain with no double bond across the path. No double bond across the path. So every carbon atom here is saturated with hydrogen atoms. Then if we come to the oils, we'll see that, so this is an unsaturated fat molecule. Okay, you see here that you have a double bond found somewhere here. Okay, this double bond at times causes this to bend. So triglycerides are formed by the condensation of one glycerol with 3 fatty acid tails. Okay, one glycerol with 3 fatty acid tails. So I have a glycerol. Glycerol is an alcohol that has 3 carbon atoms, and it has also on each of these carbon atoms hydroxy groups. So these hydroxy groups, along with the hydroxy groups that are found, remember, on the carboxylic acid functions, that is terminal in the fatty acid, are going to condense. So there will be a dehydration reaction or a condensation reaction, which is going to create this bond here that is going to link the glycerol and fatty acid. So triglyceride, we call them triglyceride because you have 3 fatty acid and a glycerol. Okay, you should note that. So 3 fatty acid combined with a glycerol to produce a triglyceride, and there is a release of 3 water molecules. So these are the commonest lipids in nature, and they are further classified as fat or oils if they are solid or liquid respectively. So fats are solid, oils are liquid. Example of fat, we give butter. Example of oils, the normal oils used in the house, groundnut oil. So they are non-polar molecules. So these triglycerides, they are non-polar molecules that are mainly used for energy production. Okay, they have a higher calorific quotient compared to glucose at an equal mass because of their higher hydrogen content. So what do we mean by higher calorific quotient? It means that the energy that your body can obtain by breaking down fat or triglycerides to produce energy, that is the energy it can obtain by breaking down this thing, is very high compared to other macromolecules like carbohydrates, for example. Why is it that your body can obtain more energy by breaking down fat than breaking down the carbohydrate? Because of the hydrogen content of fat. Fats have a lot of hydrogen content, okay? So they can produce more NAD and fat. You see that when you be doing metabolism, okay? Then next, they are located beneath the dermis and thus provide a means of insulation against heat loss. So you see this in your histology, the dermis, and special sense. You see the dermis, you have what we call the hypodermis, that is made up of a fat layer. Okay, a fat layer. And those fat there, they mainly help in insulation to prevent heat loss. And they can also serve as a support system for some particular organs. So they can help to support and also to cushion some organs. It's not written in the note, but you should note. To cushion, meaning to like protect the organ from some shock, like organs like the heart and so on. So you have here, this is a glycerol, that is an alcohol that is made up of 3 carbon atoms and 2 OH groups. Then this is your fatty acid, which we said has a carboxylic acid group that is terminal, and then you have a normal alkyl. So this one has 1, 2. This R group that is linked to this carboxylic acid has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15 carbon atoms. Okay, so this fatty acid here is palmitic acid. This is palmitic acid, the one in your palm oil. So this, what happens is that there is a condensation reaction in which this OH and H, these two OH, you have OH from one of these groups and H that combine together to form water, and at the end there is a bond, and this fatty acid becomes bonded here, and then the next fatty acid will come and bond to the next OH, and the last fatty acid will come and bond to this last OH here, forming a single glyceride bound to 3 fatty acids, so triglyceride. Then let's enter into the next, phospholipids. So phospholipids, they are lipids that contain a phosphate group. Okay, so phospholipids, they are lipids that contain a phosphate group. So here, one of these OH groups in the glycerol, remember that last OH group in the glycerol combines with phosphoric acid instead. So it does not combine with another fatty acid, but instead with phosphoric acid. So we will likely see the structure of phosphoric acid, while the other two combine with the normal fatty acid as normal. Okay, so as a result, the phosphate part of the molecule is polar, right? Polar meaning that it has a charge and it can dissolve in water, while the tail remains non-polar. So the polar part has a charge, and as such it can easily dissolve in water. Okay, so polar meaning it will be hydrophilic, the non-polar meaning it will be hydrophobic. So the polar part, we will call it the head of the phospholipid, and the non-polar part, we call it the tail of the phospholipid. So these phospholipids, they help principally in the formation of the plasma membrane of human cells. So every plasma membrane in the human cell, it can be the surface, the plasmalemma, the surface plasma membrane. It can be the plasma membrane, the nuclear membrane. It can be the membrane from other organelles. So each of those organelles, their membranes are made up of phospholipids mainly. Okay, they equally participate in the formation of the myelin sheath. So this is an example, a little diagram that shows. So you have the glycerol here that is bound to 2 fatty acids, the glycerol that is bound to 2 fatty acids, and then the last OH binds to this phosphate group of phosphoric acid, the phosphoric acid or the phosphate groups, which itself binds to another molecule of choline, but you should mainly know that it binds to the phosphate group, and the whole of this structure is polar, and we call this the head. So we call it a hydrophilic polar head. Okay, so this is a hydrophilic polar head group, and this is a hydrophobic non-polar tail. Okay, so this is the structure of the phospholipid. So many of these now are going to be formed like this. So remember, hydrophilic, they love water, so they will tend to turn, because remember, in a cell you have water exterior and then you have water inside. There's water in the cytoplasm. So the heads, they look towards where there is water. Okay, and the tails, they look inward, they run away from water. So you always have this structure of the plasma membrane. So that's a bilayer phospholipid bilayer, which you must have seen in cytology. So another structure here. So you see, this is the structure of the phosphate group. Okay, phosphoric acid, that comes from phosphoric acid. So all of this is the head, all of this here is the head, and this is the tail. Okay, then steroids. Steroids are highly complex non-polar hydrocarbon rings. Okay, so these are very complex lipids that are very different from the others. So these steroids, they are the building blocks of of steroid, the building blocks of this steroid is the sterol which is a nucleus. Okay, the sterol nucleus, which you should you should make sure to know it. So these steroids, they are the commonest of this steroid is cholesterol, which is also a part of the cell membrane. So the commonest of the steroids is cholesterol, which is produced basically by every animal. So every animal can produce cholesterol. So the cholesterol also serve as precursors to bile salt and steroid hormones. Okay, steroid hormones, they are mainly sex hormones like estrogen, progesterone, testosterone, and all of those things. Then you also have things like, so you see that you go deep into that. But now you should know that we use steroid to produce steroid hormones. Okay, steroid produce steroid hormones, and the steroid, their building block is mainly the sterol nucleus or the sterol. So they equally serve as cell membrane constituent in animals, as we said, like cholesterol that helps in the fluidity of cell membranes. So steroids also assist in the metabolism of fat soluble vitamins. Example, vitamin D. Okay, they assist in the formation of special cell messengers, so that's hormones. Then the next and last type of lipids which we are going to see are the glycolipids. So these lipids, these lipids are associated to one or more sugar units. So glyco, this prefix here this glyco here has to relate to carbohydrate. Okay, glyco has to relate with carbohydrate. So you have carbohydrate bound to a lipid. Okay, so glycolipids, they are lipids that are associated to one or more sugar units. So they serve mainly as cellular recognition proteins on the surface cell membranes. So these glycolipids, they are mainly what forms our antigens which you have been hearing of ever since you have been doing biology. So they equally assist in the formation of the myelin sheath that covers the nerve axons. That said, nice day, and next class we are going to finish by looking at the amino acids and the proteins, and also by looking at the nucleic acids. So this is we'll now enter in the deep subject. Okay, so make sure that everything that has been said today is well grasped and that you can ask your question in your groups. Okay, that's it. Nice day to everyone, and good luck to you all.